Good morning, everyone. Today we are going to start chapter 12, that is respiration of IGCSE series. So, in this topic is small actually, it's not big, and many questions being asked from here in your multiple choice and in your paper one and paper two, as well as paper three and paper four. Those of you who have taken core, you can expect question in paper one and paper three. Those who have taken extended in Cambridge IGCSE biology, they can expect question in paper 2 and paper 4. So without delaying it, let's begin with the syllabus guideline for this. So first, we'll see state the use of energy in the living organism confined to muscle contraction, protein synthesis, cell division, active transport, growth, and passage of nerves impulse and the maintenance of constant body temperature. Then we will talk about investigate and describe the effect of temperature on yeast respiration. Then we have the two types of respiration, aerobic and anaerobic. So in aerobic section, we'll see the definition, describe word. Then we have a word equation. Then we have a chemical equation. You can expect question from here. After that, in anaerobic respiration, we have describe anaerobic respiration as the chemical reaction in the cell that break down nutrients molecule to release energy. State that anaerobic respiration less energy per glucose of equation, and we'll try to understand why. State the word equation. Then we have a chemical equation. State the word equation for aerobic respiration. For anaerobic respiration, muscles is lactic to lactic acid. We don't need any chemical formula for that. Okay, so there is no chemical formula for glucose to lactic acid. Then state the lactic acid buildup in your muscles and blood during vigorous exercise. And then we'll talk about how the oxygen depth is removed after exercise, limited to continuation of fast heart rate. Then continuation of deeper and faster breathing rate and aerobic respiration of lactic acid in the liver. So all these three sub-concepts will be discussed in oxygen. Yeah. So let's begin. So respiration is quite easy to understand. So without delaying it, let's continue with the topic. So where we use our energy, which is being produced by the process of respiration, and whether it's aerobic or anaerobic. So let's begin with the table. You can see here on the right side there is a pictures are given and some terminologies are given. The use of energy in the muscle contraction, passage of nerve impulse. So the neural conduction is also going on. That is being the energy supplied by the respiration, cell growth and division, active transport in the cell, protein synthesis in the body, and maintaining the constant body temperature. So, so first of all, what happens in the respiration is when we eat food, the food is broken down into simpler form, and by the process of cellular respiration, the food molecule is transported to the sorry, by the process of assimilation, the body will, I mean, the blood will transport all these nutrients such as glucose, amino acid, fatty acids, and the glycerol, assimilated to the different each and every part of the body. After that, cells will take the glucose, and when we do breathing, oxygen will go inside the cells by the same process. And then oxygen is combined with the food molecule, which is there in the cell, and they will respire. Its chemical reaction will occur, and CO2, water, and energy is produced. So CO2 is released out again by the process of breathing. Water is remain inside the cell for further use, and the energy is being released in the body. And that energy is used anywhere in the cell, such as in terms of growth, in terms of transportation, active transportation, or in terms of muscle contraction. If you will compare the two type of respiration, that is aerobic and anaerobic respiration. Aerobic respiration gives more energy, whereas anaerobic respiration doesn't give any energy at all. I mean, they give, but it's very less compared to aerobic. And this respiration is very fundamental process, takes place everywhere in the cell. So, so it's like an individual task performed by each and every cell. And where we use it, so we use in muscle contraction, synthesize protein, cell growth, cell division and growth, enable active transport to take place, and allow nerve impulse to be generated or transported, and maintain constant body temperature. The effect of temperature on the respiration. There are several different experimental methods that can be used, and the purpose is to investigate the rate of respiration in the organism. There are so many ways. Some methods involve the use of colored indicator. So let's see how we can use colored indicator to see the effect of temperature on the respiration. So an indicator can be used to investigate the effect of temperature and the rate of aerobic respiration in yeast, and we use is methylene blue. It's the name of the dye, and we call them a suitable indicator because this will help us to to find out the rate of the aerobic respiration. So how the experiment go on is this methylene blue is added to the suspension of the of yeast cell. Suspension means a kind of liquid in which the yeasts are present, and then we put methylene blue in that. And we have kept the yeast in such a way that it doesn't damage. There should not be a damage of cells. Yeast can respire both aerobically and anaerobically. So both are going to, by both process they will respire. Though in this experiment is the rate of aerobic respiration that is being investigated. But here we are just talking about aerobic respiration. The time taken for the methylene blue to get discolored is proportional to the rate of respiration to the yeast cell in the suspension. So more yeast, the faster it will be decolored. That's the meaning. If yeast cells are respiring in the test tube or the suspension which we have taken, they will respire, and the rate of respiration is shown by the decoloration of the methylene blue. More decolor, more respiration. What all we need here in the experiment? We need yeast suspension, we need glucose solution, test tube, stopwatch, temperature controlled water baths. These are the raw material we need. So first we have taken a test tube and we have made a suspension of yeast cells and glucose, and this is stopwatch, and it will become colorless. So methylene blue is added to the solution of aerobically respiring yeast cells. The rate at which the solution turns blue to colorless, this is the colorless, will give you the rate of aerobic respiration and with the help of timer. Controlling other variables. It is important that when investigating one variable to ensure that the other variables of the experiment are being controlled. So what all things are going to control here? Number one, volume of the color dye, volume of the yeast suspension, concentration of the glucose in the tube, temperature of this of the whole thing, suspension, pH. Obviously, pH will play a major role in the suspension because if pH is changed or temperature is changed, enzyme in the yeast cells will get denatured and the respiration rate is affected. So we have to control all this. So what result you will find? A graph should be plotted by the temperature against time. As the temperature increases, the rate of photosynthesis. So, sorry, it's not a photosynthesis, it's aerobic respiration. I will change it. Also increases the time taken for the solution to go colorless is going to reduce. So this means that hydrogen are released by the reaction more easily and faster. Hence the DCPIP accept electrons more quickly until all molecules of DCPIP are reduced. This means that it will turn less time to turn from blue to colorless. So that's the reason why the solution is becoming colorless. Okay, that was the investigation part, which we have just now discussed. You can expect question in paper 6 or paper 5 from, from this. Let's go to the next subtopic. So here we are in aerobic respiration. So aerobic respiration requires oxygen and is defined as a chemical reaction in cell that use oxygen to break down nutrient molecule to release energy. And the best part of this process is there is a complete breakdown. So when we say complete breakdown, that means more amount of energy is going to produce. Larger amount of energy is going to produce. Apart from the energy, they will produce carbon dioxide and water. So equation becomes like this: glucose plus oxygen. It occurs in mitochondria. So mitochondrial cells uh, in the cells are given suit and water. So this is the equation. This is a chemical equation, and this is the word equation. What is anaerobic respiration? Here also energy is going to produce, but less. Again, food molecules are going to break down. Okay, so anaerobic respiration does not require oxygen, so we don't need oxygen here, and is defined as the chemical reaction in cell that break down nutrients molecule to the release of energy without using oxygen. So we are not using oxygen and the food molecules are broken down and we get less energy. So as I said, it is incomplete breakdown of the food molecule because there is no oxygen. So when it is incomplete, obviously it will be the energy which is being produced that will be relatively of less energy. It produces different breakdown products depending on the type of organism. So again, apart from energy, what product we will get is again not fixed. It depends on where this reaction is going on, in which cell. You need to know that equation for anaerobic respiration in human and in yeast. So in your syllabus, we'll just focus on human respiration results and yeast respiration results. Aerobic respiration in animal. Aerobic respiration mainly takes place in muscle cells during vigorous exercise. When we exercise vigorously, our muscles have a higher demand of oxygen, actually energy, actually both. Because first we start doing muscle activity. The first thing the muscles need is the energy. And when we are resting or exercising normally, our bodies can only deliver so much oxygen to our muscle cells for aerobic respiration. In this instances, as much glucose as possible is broken down with oxygen, and some glucose is broken down without it. And when it is broken down without oxygen, we will get lactic acid. We are not going to get the high yield of energy. There is still energy stored within the bond of the lactic acid molecule that the cell could use. For this reason, less energy is released when glucose is broken down anaerobically. So that's a very important point. Always remember 2 points are important here. First, there is no required oxygen. Second, lactic acid is produced in the case of animal cell. So glucose converted into lactic acid, this occurs in the muscles. So when your muscles work vigorous activity, at that time lactic acid is built up. Now let's talk about anaerobic respiration and the oxygen depth. Lactic acid built up in muscle cells and lower the pH of the blood, pH of the cells. So what is happening? Lactic acid, as the term said, acid will decrease the pH of the cell. Or if lactic acid is released into the blood, blood pH will also go down. So first of all, this lactic acid is produced in the cell. So we'll talk in terms of changing the pH in the cell. So when the pH will decrease, obviously the enzymes which are present in the cells will get denatured and they are not going to work out. So cells excrete lactic acid into the blood. So when the blood passes through the liver, lactic acid is taken up into the liver cell where it gets oxidized. Means now lactic acid is further combined with oxygen to produce carbon dioxide in water. So lactic acid reacts with oxygen. This is actually aerobic respiration with lactic acid as a nutrient molecule instead of glucose. So here lactic acid will be the nutrient molecule. Where this process going on in the liver in case of anaerobic respiration. So the waste product of lactic acid oxidation are carbon dioxide and water. This is the reason we continue to breathe heavily and our heart rate remain higher even after finishing the exercise. We need to transport the lactic acid from our muscles to the liver and continue getting larger amount of oxygen into the blood to oxidize the lactic acid. And we call them repaying the oxygen debt because you have used it in the physical activity and now it's a time to repay it. We take advantage of the product of anaerobic respiration in yeast. So in the field of baking or in the field of food industry, we are using a large amount of yeast for making bread, where the carbon dioxide is produced and makes the dough rise. And in brewing, where the ethanol produced gives beer its alcoholic nature, and the carbon dioxide which is produced will give a fizz. And this is the equation. In this slide, this chemical equation is not important. The important is the word equation. If we will compare aerobic and anaerobic, so let's see in terms of 4 discrete points. First is oxygen. So in aerobic, yes, we need oxygen, and in anaerobic, we don't need. Glucose breakdown complete in case of aerobic, incomplete in case of anaerobic. Products, we get carbon dioxide in water. In animal cell, we get lactic acid, and in yeast, we get carbon dioxide and ethanol. Energy release is larger amount, here is a less amount. So this slide is again very important. And that's it about the topic. I will try to add frequent videos on the topic as we go further. Thank you for your, thank you.